This is screen capture video 11.2 over heredity, and in the first video I finished up talking about terms, coming back and looking at Gregor Mendel's work with the pea plants. You can see the capital P, that's for parents, that would be the purple flower and the white flower. These are crossed, and the first generation would be the F1s, all their offspring would be the first generation. Those are the offspring. And as it occurs, you notice that they all have purple flowers. Even though they would have, all of them would carry the allele, the gene, for white flower color. The white flower color is recessive, purple is dominant, so purple masks over the white, and all the offspring show up with a purple flower as their trait, or that particular phenotype. The P generation, the parents were pure or true breeding, so when the purple flowered plants were self-fertilized, all that they would produce would be purple. With the white flowered plants, they were pure breeding, so when they self-fertilized, the only color flower they would produce would be white flowers. That goes along with the true breeding. So you cross the purple flower plants with white flower plants, they're true breeding, all the offspring have purple flowers. Then if you cross the F1 generation with itself, the F2 generation, which is a result of crossing members of the F1 generation with each other, give this ratio of 3 to 1. 3/4 of the plants would be purple, 1/4 would be white, and that's a 3 to 1 ratio. That 3 to 1 ratio is characteristic of crossing the F1s with themselves. And that goes along with the idea of independent assortment. When we take a look at the genetic makeup, and the genes are not something we see, the phenotype would be something we see, that's the physical expression of the genes. But the genotype we don't actually see. With the parents, the only type of gamete that the purple parent would produce would be a big P gamete, or gametes that carry a big P. The white flowers are shown with a lowercase p because it's recessive. The only type of gamete they would produce would be a little p gamete, or gametes that contain little p. So big P crossed with little p, and all the offspring, all the F1s would be big P, little p. And all that would show up as color would be purple, dominant. Well, you cross the F1s, half the gametes could be big P, and half the gametes could be little p. And since you're crossing F1 with F1, the male gametes, big P, little p, the female gametes, big P, little p. And so when you put these together, big P, you get the what would eventually be a pure breeding flower plant, big P, big P, big P, little p, big P, little p, and then little p, and because there's no dominant trait, dominant allele, in the white flower plants, the white color shows up. So the genotype ratio is 1 to 2 to 1. The phenotypic ratio is 3 to 1. 3 purples to 1 white. Let's see what this might look like if we're looking at a chromosome. This is greatly simplified, but on the chromosome that would come from the, or be in the mother, coming from the female part, you have the big P, little a, big B. If they're capital, that means they're dominant. If they're lowercase, it's recessive. In the male structures, you have the chromosome, you have the big P, little a, little b. And so an individual that would get the big P would be homozygous for the dominant allele. Dominant because it's big P, homozygous because they're both the same. An organism that would get the little a gene and little a from the two chromosomes would be homozygous because they're both the same. But since they're lowercase, it tells us they're recessive. And then for a different gene on these two chromosomes, homologous chromosomes, there's a big B and a little b. These would be called heterozygous because one is capital, one is lowercase, so dominant versus recessive, heterozygous. And we scroll on down. I was talking about independent assortment. If you use the hypothesis of dependent assortment where the traits would stay together, and this is actually a dihybrid cross where you have two different traits that are being coded for. There's the color of the grain and then its surface texture, whether it's smooth or wrinkled. And they're actually calling the smooth round. The colors are yellow and green, and then the surface, whether it's round or wrinkled. So they're showing a smooth yellow grained plant crossed with a green wrinkled seed plant. And if there was dependent assortment where these traits stay together, you would you'd see capital R, capital Y with little r, little y. And that's not what we see. This dihybrid cross is a little bit more complex than just the monohybrid cross because we've got more boxes. Because these two traits, these two characters, as we mentioned, characters, the seed color and seed surface, they're on different chromosomes. They sort independently in meiosis. And so the R's and the Y's don't stay together. So it's possible for a big R and a little y to match up. It's possible for a big Y and a little R to match up. The two different types of gametes that the yellow round seed can produce are big R, big Y. The only type of gamete that the green wrinkled seed plant can produce are little r, little y. So all the offspring would be big R, big Y, little y. A dihybrid. Then if you take these and cross them with self-pollination, the different gametes that can be produced are a big R, big Y, right here, a little r, big Y, a big R, little y, and a little r, little y. 4 different types of gametes because of independent assortment. And it'd be the same thing for the female. Big R, big Y, little r, big Y, big R, little y, little r, little y. Hopefully I said that all correctly. You can see for yourself. And when you take a look at the actual results, then you should see there are 8, 9, 10, 11, 12 yellow seeds, and there are 4 green seeds. And the colors is easier to look at. That's a 3:1 ratio. And then if you were to look at round versus wrinkle, it would be a 3:1 ratio. But when you have the dihybrid cross, you would consider yellow round, and they have the numbers here. They have the green round, they have the yellow wrinkled, and then the green wrinkled. It should be noted that you're not going to have a perfect 16, but roughly you're going to have a 9 to 3 to 3 to 1 ratio. So 9 out of 16 plants are going to have seeds that are yellow round. 3 of the 16 are going to have green round seeds. 3 of the 16 will have yellow wrinkled, and 1 of the 16 will be green wrinkled. And that's generally what we see with what's called a dihybrid cross. Dihybrid because two different characters. If we're going to use a test cross to determine the genotype, that's something that we can go back and look at dogs. There are different colored labs. There are black labs and there are chocolate labs. And the black lab would be dominant over the chocolate color. Now, if you have a black lab, you're not sure what the genotype is. You know that at least one of the alleles colors for black or codes for black color. You're not sure what the other allele is. It could be a big B or it could be a little b. Either one would still give the black color. Now the chocolate lab, because chocolate color is recessive, you know that both the alleles for that character are recessive, little b. So in doing a test cross, we take a homozygous recessive, the only type of gamete that can produce a recessive or a little b gamete, and we cross it with the unknown. This is our black lab. And the black lab, as I mentioned, it can produce a big B or it can produce a only big B, because both of them are big B's, or a big B, little b. And you take a look at the offspring. If the offspring end up all black, because the only gamete that the chocolate lab can give to the equation is a little b, if they're all black, you know then that this black lab has two big B alleles. If half the pups are black and half are chocolate, you know that its genotype is big B, little b, because it needed that little b to match up with the homozygous recessive to give the chocolate color. Here's another interesting illustration, and this comes from one of the editions of the text. The labs also can have normal vision or have a blindness. It's genetically based. And what they're showing here is that the blindness, the gene for blindness, is recessive. Normal vision would be dominant. So they have a black dog here, black coat, normal vision. We would know that two of the alleles are dominant. Big B, big N. We're not sure if there's another big B or a little b, and whether there's another big N or a little n. The dog with the black coat that's blind would be big B, not sure what the other allele would be, and two little n's, because to be blind it has to be homozygous recessive for that trait. The chocolate coat, normal vision dog, two little b's, and since it has normal vision, it'd have a big N, not sure what the other one is. And then for a chocolate lab that's blind, it would be homozygous recessive. That's the only type of gamete that it could produce would be a little b, little n. Now, if you want to know what the genetic makeup of this black lab was, it might not be considered ethical to do it, but you could cross it with the homozygous recessive, and if all the pups are black, you'd know that the other allele for coat color would be big B, and if all the pups had normal vision, you'd know that the other allele for vision was big N. And then if half of them were chocolate, you'd know it was a little b. If half of them were blind, you know it'd be little n. Now if you take a hybrid for these traits, hybrid traits, you have the big B, little b, big N, little n, big B, little b, big N, little n. Across these, you're going to get that 9:3 to 3:1 ratio. That will be called a phenotypic ratio, the physical expression of the genes. This brings us to chance events, and I'll try to tackle that in the next video.